Okay, hello bio. This is Mr. B. We're continuing on with section 2. We're looking at cell division part 2. We looked at the cell cycle last time, talked about the three of the four phases, and we're going to get into the last phase, the last phase of the cell cycle in this video, and that phase is known as the M phase or mitosis. So we're going to look at what are the four phases of mitosis, and they have divided them up into the four phases known as prophase, that's step 1, metaphase, anaphase, and telophase. So there's really no good way to kind of remember that besides, this is just one of the things you kind of have to memorize. So prophase, metaphase, anaphase, and telophase are our four phases of mitosis. So here's kind of the cycle of what it looks like here. Each step broken down, and we're going to look at each one individually. So we're going to start right off with prophase. So this is the first, and this is the longest phase of mitosis. So the first step of what happens here, the centrioles separate and they take up positions on opposite sides of the nucleus. So that is step 1. Centrioles, remember we said, were organelles that aid in cell division, and if we look over here to the right, the two almost little licorice looking things in your picture, those are the centrioles. So those are usually kind of hanging out here in the middle of the cell, and however during this phase they're going to move to opposite sides of the nucleus. And I'm sorry, I called this the entire cell. This is just the inside of the nucleus. So they move to the opposite sides of the nucleus. I'm sorry, correction. This purple area is the nucleus. This other gooey light bluish area is the rest of the cell. So first step, the centrioles move to the opposite sides of the nucleus. In the next step, the centrioles are going to lie in a region called the centrosome. So this area out here is called the centrosome, and this helps to organize the spindle, which is a fan-like microtubule structure that helps separate the chromosomes. So all these little firework-looking lines, that's what they kind of look like to me, is fireworks. Those are something called spindles, and these are just going to help us separate our chromosomes here down in the bottom of the picture. So these are organized during prophase. Then also during prophase, the chromatin is going to condense into chromosomes. So we have it right here. So usually they're kind of more loosely spread out, but since they're ready to divide, they're going to condense. And then the centrioles separate and the spindles begin to form. So again, that's just kind of a little review. And then the nuclear envelope breaks down. So that is, remember, the surrounding of the nucleus, it kind of keeps it all in, is going to break down. So that is it for prophase. It is long, but we will review it in class again. So if you have any questions, write those down. So our next phase is metaphase, and this is the second stage, and the chromosomes now are going to line up across the center of the cell. So that's illustrated pretty good there in the picture. And then the microtubules are going to connect to the centromere. Remember, the centromere kind of sounds like center. That's that little center area of the chromosomes. And the microtubules connect to the centromere of each chromosome to the poles of the spindle. So pole just being the end. So you can see we have long extensions are going out and they're grabbing right at the centromere coming from both sides of each chromosome. So that's what happens in metaphase. They get attached there. In anaphase, this is our third phase, and the sister chromatids, remember we said that chromosomes are made up of chromatids. They separate into their own individual chromosomes, and they continue to move until they have separated into two groups. So during anaphase, the chromosomes get pulled apart into chromatids is what happens. So that's what happens in anaphase. Chromosomes, they move away until they will reach the ends and get on completely different sides. And then telophase, this is our final stage of mitosis. The chromosomes are going to gather at opposite ends of the cell, and they're going to lose their distinct shape. So as you can see, the chromosomes, they've kind of formed, they're in their own little area there, and they're basically getting ready. They've basically formed two cells that are connected at this point. However, we're going to have one more final stage that's not considered part of our mitosis that will separate the cell completely. And then the new nuclear envelope forms around each cluster of chromosomes. Remember, this purple area is the area inside of our nucleus or our nuclear envelope. And so in order for this to become two new daughter cells, a process called cytokinesis must occur. So this cytokinesis pinches the cytoplasm in half. So remember, the cytoplasms are jelly filling. During this process, it just gets pinched in half. And each daughter cell has an identical set of duplicate chromosomes. So inside of this condensed area, or this purple area now, we now have chromosomes available to use. And then that's what occurs in animal cells. And in plant cells, a little bit different things happen. A structure known as the cell plate forms midway between the divided nuclei. So as you can see in that picture there, there's a distinct line that's called the cell plate. And the cell plate gradually develops into a separating membrane, and a cell wall then begins to appear in the cell plate. So that is how plant cells differ from a little bit from animals. So that is it for this video. Let me know if you have any questions.